Good morning, good afternoon, and good evening. My name is Antonia Zapata. I am a senior scientist at Nanopharm, and I'm going to present a piece of work we carried out with Diomics Corporation on the development of a nasal spray containing a human novel antibody for SARS-CoV-2 therapy. Diomics Corporation isolated and identified a novel recombinant immunoglobulin G clone potent enough to neutralize SARS-CoV-2 virus particles with 98% effectiveness. The administration of a long-lasting antibody nasal product as a prophylactic treatment might therefore positively affect the SARS-CoV-2 infection patterns at its severity. In order to achieve a long-lasting protection through the administration of a nasal product, a mucoadhesive polymer should be employed during formulation development. Chitosan is a linear polysaccharide positively charged at physiological pH. Its structure allows its interaction with mucin present in the nose. Chitosan-mucin interaction has been proven in vitro and in vivo, and its mucoadhesive properties have been exploited in order to achieve the long-lasting effect. The aim of this study is to investigate the possibility to develop a formulation suitable to deliver a recombinant antibody in the nasal cavity for prophylactic treatment of SARS-CoV-2, employing for this an Aptar BP3 nasal pump. This approach is very interesting in the treatment of viral infections in general, and innovative compared to classical intravenous administration of antibodies. However, it does present challenges, particularly related to the protein aggregation due to shear stress. In order to screen the feasibility for different delivery doses of the antibody, two formulations were manufactured at a high and low protein concentration. The antibody solution supplied by Diomics Corporation was concentrating from 8 mg ml up to 40 mg ml. The final formulations were manufactured by mixing together the antibody at two different final levels, 20 and 2 mg ml, in a buffer solution that also contained a preservative, a mucoadhesive polymer, and a surfactant to prevent the antibody aggregation. Monomeric and dimeric peaks of the antibody solutions were identified and quantified by size exclusion chromatography after the antibody solution was concentrated to 40 mg ml. The area percentage of the dimeric peak was lower than the reference, indicating that the process performed did not cause any aggregation of the protein. After the formulations were manufactured, the antibody concentration quantified was close to the target, and the area percentage again of the dimeric peak was similar to the values obtained previously after the concentration step, meaning less than 1%. As a part of the characterization of these formulations, the pH and osmolality were measured as well, in order to assess if they were suitable for nasal delivery or not. pH for both formulations was slightly acidic, which is usually recommended for nasal formulations. These values are within the upper limit of a range of pH suitable for nasal delivery, which have been observed to promote the interaction between chitosan and mucin. Therefore, it was expected to preserve the protein stability in the formulation. The osmolality measurements obtained for both formulations were inside the range for optimal values for nasal delivery too. In this way, both of them have values of around 400 millimoles per kilogram. Finally, the spray content uniformity for 10 consecutive shots were performed for each of the formulations in order to measure the protein quantification and stability. The average percentage of the target delivered dose was around 120% for each one of the formulations, with a very similar standard deviation for both of them, 5%, also very low. No protein agglomeration was detected as the dimeric peak remained at a low percentage as in the previous concentrated solution. When it comes to the spray pattern and plumb geometry, no apparent differences were observed between the two antibody formulations. However, when comparing the plumb geometry of a blank formulation or placebo with the two final formulations containing the antibody, even though they all had a similar plumb length, the plumb angle was higher, around 52 degrees, for the blank solution compared with the other two formulations. As for the spray pattern, the average droplet diameter of the final formulation containing the antibody were lower than the one collected for the blank solution or placebo. This is possibly due to the different composition and higher viscosity, which also influence in the difference of the rheological properties when the antibody was present in the solution. Mean hydrodynamic diameter of the protein was measured by dynamic light scattering, and the average values for both formulations were between 6 and 10 nanometers. The zeta potential values for both formulations were higher than 25 millivolts. This positive value is probably due to the presence of chitosan indicating a possible good interaction with mucin and moderate stability against aggregation due to charge stabilization. After loading the two formulations in an Aptar VP3 pump, the formulation device combination was characterized by droplet size distribution by Sprytek. Particularly, droplet size distribution was comparable between the two formulations, and mean volume diameter, D with 50, was higher than 50 micrometers. As final remarks, just say that this study focused on the development of a nasal formulation comprising a novel antibody for SARS-CoV-2 prophylactic treatment, and the possibility to deliver to the nose the antibody employing an Aptar VP3 pump was assessed in this process. Two formulations in this way were manufactured at two levels of protein concentration, 2 and 20 mg/ml respectively. Both formulations proved to be suitable for nasal delivery in terms of pH, droplet size, spray pattern, and plumb geometry, and also osmolality. Moreover, no protein agglomeration was observed in the two formulations prepared and after spraying with an Aptar BP3 pump, indicating favorable results in applying this system for delivery of antibodies to the nose. The next steps of the development will be the assessment of the stability under refrigerated and ambient storage conditions of the formulations. Moreover, in vivo animal studies will be carried out in order to determine pharmacokinetics and pharmacodynamics of these two formulations. Thank you very much for your attention. Please reach out if you have any comments or questions. Thank you very much.